Hello, everyone, and welcome to today's Wolfram Institute livestream. Today, we're joined with Brian Silverman, an MIT student from the 1970s and quite involved with the MIT Media Lab. Uh, Helped invent the programming language for students called um, Scratch. Involved with Turtle graphics from the beginning. Inventor of several well-known cellular automata rules, including Wire World. Worked on a precursor to Lego Mindstorms, and even participated in making a museum installation, mechanical computer made out of Tinker Toys. So basically sticks and wheels and pulleys, which played a perfect game of tic-tac-toe. Today, Brian will be giving a presentation on the machinery of life, an overview of molecular biology and how molecular machines are doing the computation necessary for life. Brian, go ahead.
Okay, well, thank you, Duga. Thank you for inviting me. Um, this talk is actually a repeat of a talk that I gave at the Wolfram Summer Research Program last summer. So it was I originally put it together to be talking to a collection of ultra bright high school kids. And what I was trying to do there was try to the majority, not all of the kids that go to that are sort of in math, physics, computer world, and a bit less so in the biology world. And I wanted to call their attention to biology because molecular biology, firsthand, I didn't like biology when I was a student. I thought it was just memorizing long lists of things and, various insane constants and things. But then in my 20s or so, I started reading Stephen Jay Gould's natural history stuff, and that led me to reading Dawkins, which led me to start reading molecular biology texts. And what I discovered in reading all of the molecular biology texts is that the chemistry there it's just really the materials, but to really understand what's going on, it's more the architecture. And the architecture is very much machinery. Now, I should say that the title of this talk, The Machinery of Life, is cribbed from this guy, David Goodsell, who wrote a wonderful book by that name. And I thought, that talking about life in terms of machinery was really a good idea. So one of the reasons, again, that I was motivated to do both that talk and this one, and don't take this too literally, is Erwin Schrödinger in the late 40s, early 50s, wrote a book called What is Life? And the way I understood what is life, it was a call out to the world's physics community saying, the war's over now. We need a new problem. The new problem should be understanding what the genetic material is, really crack the puzzle of, what's the stuff that allows evolution to happen. I think now we're at another point in history where people outside of biology should consider diving in a little bit to make it a little bit more interdisciplinary. Because biology, up until maybe a generation ago, I'm not even sure it's as long as a generation ago, the rate limiting thing was data gathering. It was very hard to gather stuff up, and there was very, very little analysis because there wasn't much to analyze. In the last 15, 20 years, we've gathered up mountains of data, and there's lots of mountains of data that there's all sorts of opportunities to be looking for patterns in that mountains of data. And maybe the result of finding some of those patterns will be to understand life a little bit better. So let me actually pull up the presentation that I did for this and do share. So um, let me do just a tiny bit of rearranging. Oops. Okay, here we are. And so we're all seeing this. So, if we're talking about life and evolution, it's always good to start with a quote from Darwin. So, what Darwin said, and this is a bit clunky because back then they wrote much longer sentences than we do now. There is a grandeur in this view of life, with its several powers, having originally breathed into a few forms or into one, and that whilst the planets have gone on cycling on according to the fixed law of gravity, forms so simple beginning, endless forms most beautiful and most wonderful have been and are being evolved. And it's I kind of picked that quote because it really makes the point that um, the stuff that we do with computational reducibility and simple programs really is something that's simple, where in biology, almost everything is emergent. So, again, this talk is intended to be a really ridiculously quick overview of some thin sliver of how molecular biology turns into machinery. So I wanted to start by saying broadly, what kinds of structures do we have in life? Um, this is the Six Kingdoms view. I know that the people who do taxonomy for a living just get into an ongoing fight about how many kingdoms there are, and there's super kingdoms and sub kingdoms and whatever. But the Six Kingdom view, for some period of time, was a consensus. Now, where this started is back when we first started naming things, there were plants and animals. So there was the plant kingdom and the animal kingdom. And they're all multicellular and they're eukaryotic, which is a word that I'll explain on the next slide. And after a while, they noticed that plants and funguses aren't really the same thing. So they added a third kingdom, the fungus kingdom. It took a while that they realized that there's stuff that you could only see through a microscope. There's unicellular, real little guys. And that ended up being called protista. And it was only when the microscopes got better that they realized that there was a different organization, which was bacteria. And bacteria are much, much, much smaller and much, much, much simpler than the eukaryotic stuff, which again, I'll show you in the next slide. And only relatively recently being about the 70s, they realized that bacteria, there's really two kinds that are dramatically different. There's the old guys and the new guys. And the connection between these two is looser than the connections between these two. So, the archaea are, these archaea, we didn't find them that soon because archaea tend to be living in crazy places like ocean vents. We found a few on drill bits that were drilling a kilometer down to find oil. Um, oh, by the way, I said it was the very next slide after. This is an overall timeline of when life came up, and it goes back 4.5 billion years. Now, when I first saw this number, this 4.56, I found it really, really confusing because the core formation and the earliest evidence of fossils date to around the same time, which means that life appeared like an instant after it was physically possible for life to appear here. The thing to notice in this is us people represent almost, it's a blip in late evolution. And even the dinosaurs are not as old as that. Now, um, in looking at this, by the way, what all of the things here are is this earlier ones is where they found things, and some of the later ones are have some shapes of things. This period here is really, really interesting because prior to here, the atmosphere of the Earth, the Earth's atmosphere is about 75% nitrogen, three-quarters nitrogen. The fourth quarter used to be carbon dioxide, and now the fourth quarter is oxygen. In a short period, the early life expelled this toxic, horrible stuff called oxygen. And oxygen ended up taking over the atmosphere, which led to a new kind of life, which was used oxygen as the main thing that it needed rather than carbon dioxide. Now, one of the things is what's an interesting coincidence is the energetic efficiency of life is about a dozen times greater in an oxygen atmosphere than in a CO2 atmosphere. So what life did is life evolved in a way that it re you can't use the word terraform for the atmosphere. It just reworked the atmosphere so that it would be better for more efficient energy management. And everything post that is kind of like the oxygen world. Now, When I was saying I was going to say something about eukaryotes and prokaryotes, when I was first getting started on this, a thing that I wanted to have, and the books tend not to do that, is providing a size scale. Because when we're talking about animal cells and plant cells, they're really, really big. Bacteria are like, oh, I don't know, a 20th a 20th the linear dimensions. So, yep, a thousandth or, 10,000ths of the volume. And even smaller than that are viruses. So, um, nothing bigger than this really happened for the first few billion years. But then after the oxygen catastrophe, there started being these big complex things. Now, not only are these things bigger, they're spectacularly more organized. That what's inside of bacteria is more or less soup in a bag. And the contents of the soup and the nature of the bag are really intricate and really interesting. More details later. But the eukaryotic thing, the cells in us and in plants, it look almost like a community of these things that built a city around themselves. That this thing, which is a mitochondrion, is the same size and same organization of a typical bacteria. So it's almost like a bunch of bacteria got together and decided that instead of having their own genetic material, they'd have a big warehouse of shared genetic material. So this is what a typical bacteria looked like. Now, I gave David Goodsell credit for the machinery of life. He's a biologist, and what he does as a hobby or as a second job is he makes these beautiful images of biological stuff. And what this is this isn't a photograph. However, he wanted to do something that was of photographic precision, but more aesthetic. So what he basically did is this is fairly accurate. If you looked at the thing under a microscope, you'd find that the coloring is all off. A little bit of the relative proportions are all off. He colored things and made things stand out. Um, in a way that tries to give some idea of what's going on. But the thing to notice here and more later is there's a tangle of string at the center of it, and then there's a soup of chunks around it, and then there's a container for the whole thing. Now, molecular biology started in around the 70s. Watson and Crick started in the 60s, really came of age in the 70s, to be a bit more accurate. Um, After Schrodinger wrote What Is Life for the search for the genetic molecule, um, people kind of found it within 5 or 10 years, but it took about another 5 or 10 years for Watson and Crick to start and figure out what was the nature of the molecules. And what they discovered very early on is a thing that it was Francis Crick, who made up the central dogma. Now, he was a bit of a playful guy because he didn't really mean dogma. He basically said that there's this unbreakable rule that he decided to call it dogma just so 20 years later, when it was broken, people could laugh about how it wasn't as dogmatic as after all. Now, what it was is DNA. DNA is the string here, um, gets transcribed into RNA. RNA is similar to DNA, but it's only like half of it. DNA is a famous double helix, and RNA is more or less one of the helices. It's got a little bit of a modification to it, so it's not quite right. RNA is basically a sequence of bits that gets translated into proteins. Proteins are actually the important stuff. And the thing to take from this is DNA is really code. It's a linear string that codes for a three-dimensional object. And I'd like to go through, some of the detail about how that linear string actually codes for an object. But first, this is a zoom in of that earlier good cell picture, where what you could see here, and here I'm going to be pointing to lots of little things, on the DNA strings are these little machines that do the transcription. And these little pieces coming out of those little machines are the transcriptions in the form of RNA. The RNA then finds their way into another machine called a ribosome. And that ribosome reads the code off of the DNA and produces a protein. The protein starts off as a linear object, but it falls into a complicated object that has a lot of different uses. Some of it is rate control for reactions. Some of it is almost more machine-like. This is a bacterial flagella. Okay, that's kind of like a tail that whips around and looks like a propeller. This thing here um, is made out of proteins. It's made largely out of proteins. It's got a few other atoms there. The collection of proteins actually form what it looks and acts so much like an electric motor. It's probably fair to say that it's an electric motor. So somewhere on the string here is the coding for a dozen or so different strings that get turned into different proteins that self-assemble into an electric motor. Now, what are these proteins? Now, here's another thing that Goodsell made a nice wall chart that let me actually zoom in a little bit on this. Um, this is just sort of a quick tour of different kinds of protein families. One of the things to notice is a lot of proteins live inside of the membrane. And I'm not 100% sure if I wanted to make a wild guess about why all of these proteins live in the membrane, is it makes them meeting each other a two-dimensional problem rather than a three-dimensional problem. And what we have here, these things are enzymes that help digest. These things are blood plasma. Um, this particular one, this giant one, this is the protein that self-assembles into these tubes that assemble and disassemble and provide structure for the eukaryotic cells. The eukaryotic cells hold their shape because they have something that's more or less like a skeleton. And by the assembly and disassembly of these tubes, the shapes are controlled and maintained. This particular protein is crazy. This is a linear motor. This guy has the ability to walk down this guy. And there are various movies that you can find on the web that do it. This is another one that we'll be looking at in greater detail later. This is a different electric motor that's actually being used to deal with the energy currency, and it's actually creating the thing that is the main energy currency in life. The thing that I was telling you about one machine doing copying and then doing transcribing and then turning into proteins, part of that process is in this orange and green set of things here. Um, so here, let me get this back down to 100% so we can move on. Um, So in order to show this in greater detail, if you remember from the earlier pictures, the real little guys were the viruses. So this is another good cell thing of a real little guy. This is a virus. This is our good nemesis, SARS-CoV-2, the virus, which made us all sick. Actually, I didn't get COVID. I don't know if anybody else managed to um, escape it. But so this is the good cell rendition of it. This is a somewhat different rendition of it. What a SARS particle is basically it's genetic material, it's RNA in a bubble that's created out of lipids with a few proteins that help control the thing is. These proteins, as I'm sure during the pandemic, everybody was talking about spike protein, so the spike and its mutations. The spike thing is kind of like here. The reason it's called spike is you actually look at these things, it kind of looks like it's a spiky sphere. Um, the RNA stuff here, the little green dots are basically bubble wrap for the RNA because RNA is fairly fragile. So um, the RNA here, it's not a lot of code. There are 30,000 base pairs. Each base pair is 2 bits. So there's about 60K bits that describe this. What those 60K bits do for a living is they encode for, uh, well, if we went back to this picture, there's an envelope protein and there's a spike protein and there's a membrane protein. All of the proteins that it codes for is on this side of this. The other side of this is it's little bits of mRNA that encode for little bits machinery that it is itself made out of mRNA. And um, what it was is this particular image is taken from the Proximal Origins paper, which had a lot of notoriety. That basically was saying, guessing that the virus had a natural cause. The thing about this is, let me do another zoom in. Hang on. Yeah, I actually don't think I could zoom in more close on this one. Sorry, for some reason, some images zoom and others don't. Some mathematical guru could say why. What this is basically doing is it's showing a little piece, the important piece of the spike protein in human SARS, in a bat thing, in a pangolin thing. And if you actually compare the humans to the pangolins, they're almost identical. Um, there's a place here where the spike protein is actually broken into two, and the joint between the two sites is very different in humans than in other things. And for people who are following the craziness around the molecular biology of COVID, this is the furin cleavage site that a lot of ink was spilled over. But let's get back into the detail. This is a close-up of what the transcription process looks like. There's a little machine here that the machine crawls along the DNA. And as it's crawling along the DNA, it's spitting out the appropriately transcribed RNA. Once the RNA is transcribed, this is a ribosome where the mRNA string is working its way through the ribosome, and the ribosome is emitting pieces of protein. Now, what I wanted to do with this, yeah, I'm not even sure it's worth doing, but I think I'm going to try anyway. Um, when I was saying that there's a lot of information around, there's the National Library of Medicine and at NIH has a database called GenBank, where the GenBank database has basically the genomes of um, millions, I'm not even sure. It's more than millions. It's tens of millions or hundreds of millions of different genomes. And what I did during the pandemic, just kind of for fun, is I I wanted to see how SARS was evolving during the course of the pandemic. So I was able to basically, by the way, this is just a Mathematica notebook with nothing special and is pulling the data with URL read. And the rest of the code is to be parsing the data. So I'd asked it to fetch the genome for this. For some reason, people who do biology databases always have crazy numbers to describe things and crazy names to describe things. With1 um, was the biologist name for the wild type of the SARS thing. It was the Wuhan Institute of Virology. So the first recorded sequence of SARS was done from the Wuhan Institute of Virology. And I could ask inside of the record is what genes were there, and there's a list of genes. And the spike protein at this point was called the surface protein. So to get a sense of it, the spike protein of SARS looks kind of like this. So what I did is here I was looking for Omicron because I did this during the Omicron era and looked at the Omicron surface protein.
Are you
And I
are you
said, how
showing?
different?
I think you're not showing your whole screen.
Oh, I'm not showing this. I'm not, you should have told me that earlier. Okay, share. Yeah, I actually thought it would follow on that one. Okay, share. Okay. Um, the only thing that you've seen it now, yeah, yeah.
Yeah.
Um, I just said let's get the original wild type, and what's the code for the spike protein of the original wild type? And it's this. Now, the thing about this is in the big picture, this is a lot smaller than, say, the source code for Mathematica, right? This is a relatively tiny piece of code.
So
So
what are
and
we
then
looking at
and
though? It's not genetic code. It has too many letters.
what it is it's protein codes.
Okay.
Is each triple gets contributes a letter. Okay, and get back to the detail of that in a minute. So if I got the Omicron one, and if you look at the Omicron one and the wild type one, to your eyes they look pretty similar, right? But I did comparison between the two, and you can see that there's about 30 mutations between Omicron and the wild type, right? For comparison, there was only about 10 mutations between the wild type and Delta. And here's the thing that I found surprising. So it's about 10 and it's about 30. Um, BANAL 2052 is a bat from Laos, which is the closest equivalent to the pandemic virus. And when I did the compare from the wild type to that bat, it was closer than Omicron was. Now, I don't know what to make of that, but the notion that there was a circulating bat virus that was closer to the pandemic virus than a later version of the pandemic virus, I thought that that was kind of interesting. But I'm gonna stop share and reshare it to not make the same mistake twice. Here, share. Okay. No, this isn't the one that I meant. Stop share and here, share. Share. Okay, that's interesting. It's not doing the right thing. Um, do you have any sense of why this is failing? Mm,
Not sure. So I mean, before it seemed like you were sharing the window of your notebook,
yes.
presentation notebook.
Yeah. And here, give me a second. Let me go back to tab. Here, back to Zoom. Share. Okay. And why don't I just share the desktop, which is always scary, but we could do that anyway. Okay, now if I from here I could just go back to here. Okay, yeah, this looks right. That shouldn't have been necessary. So we looked at DNA from a just string in a soup point of view. What this is it's looking at it a little bit closer. Now, um, doing this completely within the Wolfram molecular biology world is, I'd say, give me a biomolecule and I have that biomolecule come from the protein data bank. And when I was saying earlier that these databases have crazy names, the crazy name for a DNA thing is this. For anyway, at saying do it as ribbons. And what this is it's a little bit of DNA. Now, what you could notice is here you could see this is a base pair and this is a base pair. Now, one of the things, and there's going to be, a little bit of that for the next several minutes. Um, I did a bunch of consulting for Lego, so maybe that caused me to think about things in terms of construction kits. There are four molecular biological construction kits. And I'm only going to be talking about two of them. The two that I'm going to be talking about are nucleic acids and proteins. The nucleic acid construction kit only has four members. And Watson and Crick's amazing discovery was you can take this guy and bind it. If number one and number three form the same shape as number two and number four. So you can have any of the four base pairs here, and there's a matching one that allows you to have the exact same physical structure. So you can have this double helix, and the double helix is pretty much the exact same shape, independent of its bit content. So the way information is encoded, the way you encode the shape of a protein, three of these guys in a row contribute one letter to that long letter string. And the three in a row is the way in which it contributes is it's what we ended up calling the genetic code. So essentially here, there's a first row, then a column, then the third one is pick one of the four of them. So three base pairs pick an amino acid. Now, here, let me say something quickly, and not enough information, but maybe some people will stick. If you look at this, it's kind of curious that for it to be a proline, CC and don't care. For it to be a leucine, it's CU and don't care. For it to be an alanine, it's GC and don't care. You only do care in a small number of cases. Now, what Crick theorized, this is a crazy idea, so rather than me saying I'm saying it, it's much better to quote a Nobel Prize winning person. He said maybe the genetic code used to be a two code rather than a three code. And the third one only got added later on in the game. So it's kind of like the genetic code was happy with 2 gives you 16, but there are 20 amino acids, and 20 is a few more than 16. So you had to ha add a whole other base pair just because you overflowed the earlier set. Okay, now um, each of these is an amino acid. Again,
Which
just
ones are amino acids? So there are
um,
four which four amino
th-
acids used
no, there
for
are four nucleic acids.
nucleic acids.
Yes, and
Are,
there are
are those not
yeah, and
amino acids?
no, nucleic acids live inside of either mRNA or, well, the nucleic acids are the major component of the genetic stuff in us.
Mhm.
That mo most of the information processing has a um, the nu lots of nucleic acids, and there isn't that much nucleic acids outside of the genetic processing. I'm saying that, and don't take that as really true. That's more a tendency. What it is three of these guys encode for an amino acid. Now, there are 20 amino acids, and this is a list of what they look like in three different forms. Okay, the three forms, and by the way, since this is a Wolfram Institute thing, I, it's trivial to pull this up because molecule plot 3D, I'm basically just plotting the same thing with three different plot themes. So one of the plot themes gives it what looks like a chemical formula. The second one is ball and stick, and the third one is space filling. These are all the same thing. Now, what's interesting here is the way proteins work is we can go through them. They all share, you can see O, weirdo bonding, H. You see it here, you see it here, you see it here. You see it here, here. And they all have this NHH. Okay, so essentially this piece, this and this are kind of a backbone. And added to the backbone is a little additional thing. And that little additional thing is what, as the thing folds up, it confers a particular shape on it. So if you look at this, the thing that confers the shape is called a side chain, and with the bigger ones, this whole thing is this. Yeah, this whole thing is the side chain. In this one, this is the side chain, and what it does is, at the end of the day, it makes this building block. It makes another construction set, and it makes a construction set built out of all of these weirdly shaped blobs that when you pull them all together, they form a three-dimensional shape. And that three-dimensional shape does something magic like catalyze a reaction or, give you a piece of a motor or allows some collections of these are used to decide what can get through a membrane or not. And basically, what all of these proteins are doing is they're basically the regulation mechanism of the chemistry that's going on in the cell. So,
Can I ask a quick question?
sure.
Okay. So you said there are 20 amino acids, but why aren't there 64 if there could be 64 based on those the nucleic acid?
Watson and Crick puzzled that question a lot. And their conclusion, which it's in fact, I kind of said this before, but not in the way that it answered your question. Their guess was the reason there wasn't 64 is that there was originally 16, and 16 wasn't enough. So when the space got increased, it got increased by it got quadrupled, but there was no real reason to quadruple it. There was only a few more added in.
So nothing kind of wanted to kind of fill in and expand that space of possibility.
Um, well, to a first approximation, nothing. Okay, because doing that would require changing the genetic code. That by definition is a change in the genetic code. And um, when the genetic code first got cracked, they thought that the genetic code was universal. Okay, but I'm actually going to scroll back a lot, and it's maybe annoying to do that. The genetic code is pretty much universal across this, and this kingdom. Okay, it's very, very close with this kingdom. This kingdom, the archaea have a genetic code that's modified. Of the 20 things, there's sometimes as much as 4 changes. Usually those changes, I could get back to the thing there. Oh, by the way, when an interesting clue, and this is a bit of a digression, the ancient guys, the genetic code of these guys. Um, Mitochondria, which are symbiotic organisms with us, have their own genetic code, and their genetic code more resembles the archaea than anything later, which is it's curious to say the least. Does that adequately answer it?
Yeah, I think so. I'm still kind of curious about like where there's room for emergence in life and why more amino acids wouldn't emerge from this new structure that was afforded by the fourth um, nucleic acid.
Um, would it be okay if I went through the rest of the presentation
Yes, of course, of course.
and then return to that question because that's a fundamentally important question. So the thing that I was trying to get at, I picked a random molecule, glucagon, only because I was looking at that chart and where's the simple one, and I was able to get a biomolecule version of it by just knowing what its magic code was. So get a Wolfram biomolecule, and then ask it to what's the sequence. Now, it turns out that this molecule, these are abbreviations for the amino acids. So glucagon is just a list of, I could do a length of this, about 20 amino acids. And if I plot the molecule as a ribbons plot, this thing folds into this nice spiral. And if I looked at it as a space filling thing, then it actually fills a lot of space and is fairly complicated. And if I do it as a ball and stick thing, I'm only zoomed in on this, but this ball and stick thing, you could see it's the amino acids from this table. And when they get joined, the way these things get joined is this and, here, I'm going to get it wrong if I don't have a simpler one. Um, Part of this gets snipped and attached to here to do an end-to-end connection. So the things get end-to-end connected and then fold into this kind of spirally thing. So what I did here actually, and I'm not even sure how much more information is there. There was the list of all of this, and just wanted to say if you looked at the same list but looked at it in ball and stick form of amino acid, what does it really look like? And you can imagine that there's a little cutting and pasting going on here that will give you exactly this. Okay, this is a bit of switching gears. When I was saying at the very, very beginning that I got fascinated by the machinery of it, a piece of machinery that I hadn't really looked at closely until the last year is how biology deals with energy. And um, what I was kind of interested, stunned, fascinated to find there are two molecules, adenotriphosphate and adenodiphosphate, where this version of it, if you look at this and this, they're identical, except this guy has three of these P things, and this guy only has two of these P things. Where in this view, you could see there's a string of three of them at the end. And this thing is a string of only two of them at the end. Or in this view, you could see that this goes on for three of them and this only goes on for two of them. And the thing that's being added is inorganic phosphate, which is a big name for this thing. And what I found fairly amazing is the way cells power themselves. Everything interesting um, is powered by breaking off one of these guys. You break off one of these guys, and it releases an absolutely nanoscopic amount of energy. But it's that energy that's being used to power almost all of the machinery. So the way that the overall power supply works is it's a very, very, very complicated thing where metabolism gives you a highly energetic molecule that then goes through this incredibly intricate set of reactions that at the end of the day um, set up a proton gradient from one side of the membrane to the other. And that proton gradient is used to power that magical molecule that I pointed before that adds one of these inorganic phosphates to here. Now, um, what's really going on here? And again, one of the reasons I was thinking of this is machinery of life. Establishing a proton gradient. What a proton gradient means is there's more positive charges here than here. Me with my electrical engineering background says that the name for that is voltage. So basically, the stuff that you eat is being pushed through a complicated piece of chemistry that establishes a voltage, and then that voltage is used to turn an electric motor. Here's the electric motor. What the electric motor does is this turns, and as it's turning, there's a camshaft that camshaft is pushing this guy onto this guy to make this guy.
Okay,
So
so it's an endothermic reaction, adding the extra phosphate on that chain of the diphosphate to the
yes,
triphosphate, right? So
yes, and it's
physically, like
basically
to put that thing on, it's like magnets, right? So you have to like push it on with a certain
it's
amount of
like
force,
yeah,
and then when you snap it
yes,
off, you get that all that energy back. So these things go flying apart kinetically.
yep, yep, and you get that back,
Hmm.
and these things, the thing that's pushing it is this spinning, and this spins at hundreds of times a second. And um, here, I should say this carefully because it's easy to confuse. These guys get reused, right?
Uh-huh.
That this gets turned into this, gets turned into this. It cycles and it cycles real quick, like in the seconds to minutes time scale. Okay. But if you just added the amount of, if you added the weight of all of these guys together, even though they're reused, so it's a bit of double counting, it's equal to about your body weight per day.
That it
So
metabolizes.
yes, the mitochondria have many, many, many of these things. So, this whole process is what I found very, very, very fascinating is just like a good power supply designer,
Mhm.
um, the um, the voltage rails, the actual source of energy that the whole machinery is using is consistent and independent of where it came from. And what it is that's providing the proton gradient is it's plants do it a bit differently than animals because in plants some of this is being done through photosynthesis. Stuff that some of the archaea is doing this in a pre-oxygen world. And the thing that I was saying about things having gotten much more efficient, the oxygen finds its way into here. And um, essentially, this is a simplification. This is dropping an electron down a big tunnel. As the electron is finding its way through the tunnel, it's pushing protons across the membrane. And the place where the electron ultimately lands is onto an oxygen. So the role of oxygen in this is the ultimate electron acceptor. In archaea, where the ultimate electron acceptor isn't oxygen, you only get about, I'm going to get the number wrong, but I'll make it up. You get about four push magnet pushes per electron. With the oxygen acceptor, it's not four, it's more like 20.
Oh, so the reactivity of oxygen is what's giving this uh,
Well, it's not really the
benefit.
reactivity of oxygen. It's the fact that oxygen, an O2 molecule, is what's the right word? It's sufficiently low energy that anything high energy has to fall a long way to get to there. Oh, okay, that yeah, I'm not quite sure what the right way to say this is the electrons can could be used more times on its way through the tunnel. If the ult it's sort of like you can imagine that you're dropping a ball off the roof. If you drop it to a porch on the first floor, you're not going to get as much energy as if you drop it to the ground. And oxygen is closer to the ground than the other electron acceptors that naturally occur.
I see.
But okay, anyway, this is an artist view or a CGI view of this machine. This is the biomolecule plot 3D view of this machine. And just to get a sense of it is when I was saying that ATP is the energy currency, I just made a little table. What's the costs of using things? So you want to add an amino acid, it's 4 ATPs. You want to do the translation of a protein, it's 1200. You want to transcribe a nucleotide, it's one. One nucleotide of replication is one or two. tRNA charging is one. Ion transport is one. Calcium export is one. Glycolysis is that's going the other way. It's the um, the citric acid cycle is pulls together 30 to 32 of them. Motor proteins and vehicle transport is one of them per 8 nanometers. Turning that bacterial motor is 1,000 of them per revolution. Chromosome segregation and meiosis is, a million to multiple millions of them. Cell division is this. The thing to notice about this is almost every energy using mechanism in a cell, the form of energy that it's using is exactly the magnetic attraction of this to this. So essentially, the way the whole energy system works is through some very standardized thing, which it's again repeating what I said already a couple of times in the talk, is it's got a level of cleverness that's fairly surprising. Now, One of the things that I wanted to say, and this didn't with the high school kids, they didn't know what I was talking about. Um, we're talking about computation, right? And we know about Turing machines and we know about universal Turing machines. One of the things that von Neumann suggested is in order for Darwinian evolution to be a thing, you have to have a universal constructor attached that is being driven by tape-based replication. And I didn't draw this. This is somebody else's trying to make a diagram of what they thought von Neumann meant. The tape is, by the way, von Neumann was talking about tapes. 15 years before the molecular biology revolution, where we knew the DNA was almost literally a tape. Von Neumann said that you should have a tape that is the description of the thing, and say that the tape gets used in two ways. One way is the tape unit is just the tape replicator. The other use of the tape is interpreted to drive a universal constructor, and the universal constructor makes more copies of the machinery. Now, von Neumann kind of suggested this, not really realizing that the whole DNA polymerase world is exactly this, and the translation transcription world is exactly this. You know, so, the question is, what von Neumann basically said is in order for Darwinian evolution to be effective, it has to be operating on a bits-based description of the structure. Because if it's a bits-based description, it's easier for mutations to be remembered generation to generation, and it's easier for things that have gotten better to be remembered generation by generation, and just ratcheting things to better and better. Now, this is the end. I just wanted to end with just an image of, you know, it used to be the tree of life. These days, people aren't happy in referring to the structure as a tree because in fact, it's a graph and it's a bit of a big mess. But even so, this was kind of an in-between kind where it was sort of tree formed. So that's kind of what I wanted to present. Would appreciate. I'll stop sharing so I can look at questions and comments. Um, is the silence because people didn't
I was just
understand a word of it?
reading this comment from
Overwhelmed?
Pedro. You know, on the subject of why there aren't 64 amino acids, basically. So you have those four codons, four nucleotides,
Yes,
nucleic acids, coding
yes,
for in triplets, all these
yes.
amino acids.
And there's a lot of redundancy. And whether that redundancy was accidental or intended is hard to guess.
Right. Can we look at that chart again?
Sure. Let me do share. And I'm going to share. No, this maybe, hang on a second. Stop share. I'm not doing the right thing again. Sharing. Yeah, desktop one, share. And then click to this. Okay. So if we looked at the chart again. Now, and this may be further answer to what Ellie was saying. In certain, the number of valid, the number of valid subset of the 64 choices is organism dependent. That because there arginine, there are four of these. Um, some creatures validly accept all four, some fewer, some one, some two, some three. Because in order for this to work here, I can go back to this picture. This sketchy molecule is transfer RNA. What this is it's a molecule that on one side of it has an anticodon. It's the thing that skips to the relevant base pair, and on the other side of it has the amino acid. So
Right, right.
each of
So
the
there are
each of the
amino
six.
acids floating around, and then there's this ribosome reading the RNA in triplets,
Yes,
and then it
yes,
reads a triplet of them,
and
and then there's some amino acid
well,
which comes in and is able to.
okay, but to more directly answer what Ali was saying is if you wanted to change this table, you'd have to simultaneously evolve a tRNA molecule. You'd ha have to build a new kind of tRNA molecule. There's a very complicated enzyme that attaches the amino acid to the tRNA molecule. So you'd have to build this guy and its larger cousin, and also insert the new use of it into some genomes. So it's kind of like three things have to happen simultaneously for a new position to change or for a new
Yeah, but
position.
we're talking about billions of years. So
Yeah, but
those things,
this is an interesting comment. We're talking about billions of years, but billions of years, billion is only 10 to the ninth. And
mm,
why don't we say 10 to the 20th, 10 to the 20th when you get it down to fractions of a second. And the universe is only about 10 to the 40th or 10 to the 60th big.
mm.
So if you have a thing like this, um, even this little thing is 60 bits, 2 to the 60th, and this is a tiny protein. A substantially sized protein is hundreds long, which there isn't enough, even if it's billions of years and microscopic, there's not enough entropy in the universe to randomly find it.
Yeah.
Right? That
Yeah,
no,
another thing, yeah, what
yeah,
Pedro said about redundancy too kind of strikes me because I bring everything back to my very simple understanding of like the 8-bit elementary cellular automata, and there are a lot of different CAs that make, say, a checkerboard pattern.
yes.
Um, but yeah, so if you were trying to achieve checkerboard pattern, there are a lot of different ways you could do it, but it's the checkerboard pattern in some sense that is the observable output. So maybe it's a similar situation in that.
It's possible. It's possible, but at the risk of being very, very controversial, um, I think what's going on in biology is it's the amount of information is too big for very much to have been found by exhaustive search. That it is the case that there's a lot of different ways of doing things, but it's kind of like it wouldn't be. There's the old, hurricane through a junkyard making the space shuttle thing, right? People who are looking at this make compelling arguments for how the amino acids, nucleic acids, could have spontaneously occurred. The argument is much less good about how a ribosome or the tRNA, how the genetic code could have occurred. And
Yeah, and
you know,
there's probably
and
also some sort of a gradient of like when you have a mechanism that's working, it's probably optimal to build on that rather than to have kind of um, nature, whatever is driving this exploration, um, kind of discover new basic elements if it's better for it to develop the machinery that's already been
right.
devised.
No, no, but for me, the fundamental question of this, if you have one of these guys, you can imagine a step-by-step process with that goes from here to Mathematica. Okay, because this guy could slowly and steadily Darwinianly turn into this. This guy could slowly and steadily kick off neuroscience. Neuroscience could kick off the computer, we could figure out how to kick off the computer industry and then things can evolve. To me, the thing that's much, much harder to get my head around, how did we go from the Big Bang to this guy? Because it's again is the argument where I think I'm actually siding with von Neumann. Once you've established a von Neumann replicator, you could do anything. Evolution could take you anywhere from a bit space replicator. The question that I had is how did the bit space replicator get there? Now,
Olga on the live stream is asking on the subject of the amino acid coding. Is redundancy meant or used for error correction?
it's not clear that it is. Okay, that one of the things when I was saying that there's amazing kinds of machinery is if I go back to um, I don't actually have the
So,
DNA replicator here.
I mean, this is something that Pedro was mentioning is that the ambiguity of the genetic code gives it more resilience.
Yeah, it's not
And
obvious that it's
it's
not
robust
obvious.
to single point mutations. So if like the third letter on one of these sequences, for arginine, CGU.
Right. No, but I could make. Could I respond to this with a crazy way? If it was my job to make a redundant genetic code, it wouldn't be this one
Mhm.
because this one is completely blown away by the change in the first or the second, and it's
Sure.
true that it's resistant to a change in the third. But if your goal was to make the thing more robust, you would have spread the redundancy across the three of them.
Yeah, in fact, maybe it was set up to optimize like um, identifying where there were errors to throw out like certain kinds of um, combinations that are not
Well,
wanted.
without going into a lot of detail of it, there is fair bits of error correcting at very different levels. And here, let me um get back to this. A thing that I learned during the pandemic that I thought was really weird. Um, one of these guys, one of the molecular machines that's built into a coronavirus is an RNA to RNA copy machine. And, basically, one of these guys is a piece of RNA that knows how to copy RNA. And I know when I first read that, I wanted to like write to that machine saying, "Didn't you read the central dogma? Because that's not allowed." Um, but viruses seem to break a lot of the rules. What's interesting about it is one of these guys, I don't remember which NSP was, is a simple error corrector. So it gives an overall replication fidelity of a given size. There's a formula that's been tested across the viral world where the replication fidelity leads to the maximum available size. And because if you make the better fidelity means you can copy bigger strings without getting yourself into trouble. Now, it turns out that in a very general way, not true in detail, the structure of the polio genome resembles this. Only polio doesn't have the coronavirus error replicator, so it can't be as big as 30,000. It can only be as big as 7,000. So the fact that the coronavirus could be bigger gives it more room to do more. And if you actually spend some time poking into the genome, the more that it does is figures out more and more devious ways um, to win the arms race with the immune system. But out of curiosity, since we were talking about fidelity, the viral fidelity is about 2 or 3 orders of magnitude lower than the DNA replication fidelity.
Which fidelity is lower?
The viruses. that it's the fidelity of genome replication in us in DNA-based creatures is remarkable. And the thing is I showed that, the ATP printing press, that particular molecule, that particular machine is universal across 99.9% of life um, that's living now and universal going back, a couple of billion years. And the thing that I thought was just like a really, really interesting question. So your job is to design a way to preserve a pattern across billions of years. What do you do? And the idea that we have particular patterns that have been preserved since, the beginning of the Earth, at some level, it's a stunning fact.
Right, right. So you made in previous conversations that this preservation is empowered by the digital nature of the DNA molecule, of these
Right. And I
codons
said,
being
yeah,
discrete letters.
yeah, yeah. And I did say, but not the same audience was there at the time. Um, Jeff Hinton was trying to figure out how to make LLMs more energy efficient. And at some point, he figured that if he went analog, it could be a lot more efficient. But then realized if he went analog, he'd be suffering the error catastrophe of not having the replication fidelity that you needed. And he figured that the only way to keep LLMs robust is to keep them digital.
To keep them consistent from one copy to the next. Yeah.
Well, not only to keep them consistent from one copy to the next, but to allow them to move from one system to the other without having to be recoded. So anyway, the idea that we're based on a digital-based, bits-based von Neumann replicator, I think is really fundamental. Now, when I was saying that there's room for a lot more work in this, everything that I showed was completely light on details. And in every corner of everything, there are details where we currently have mounds of data that may provide some insight.
Yeah, what do you think are the most fertile areas for investigation, especially by somebody who has kind of a fascination with bits-based analysis?
Well,
Um, yeah.
well, why don't I not answer this for the general someone, but answer it for me? The first exploration that I did is there are 10 million SARS genomes online. I downloaded them and then try to track mutations. Right, because the SARS genome data set is probably the best data set we've ever had for seeing evolution in real time. Okay, that's one. The second thing, which I think I'm more interested in now, is there's this emerging field of systems biology where everything that's going on is, Bob, you heard me say this before, it's a rat's nest of interacting feedback loops. And there's starting to be good databases of the various feedback loops and trying to figure out, whether or not there's any rhyme or reason in how things are working. And I could here a speculation that only has a 5% chance of being right. Um, when Dugan was going through what I'd done in the past, one of the things that I'd done in the past is emulations of old computers to get old bits of software running, and we went from there to emulations of old microcontroller chips. Well, also to just see how the old microcontroller chips work. When we're emulating chips, we found, much to my great surprise, you could ignore all of physics and just emulated as if transistors were ideal switches. And at first, it was surprising to me that that really worked, that you could ignore capacitances and resistances and inductances and just have the thing be truly binary. But then realized that the people who designed the circuits were designing them as if they were perfect. And the fact that they weren't was a bug, not a feature. So here's the speculation. Um, could we ignore pretty much all of the biochemical details of the way these reactions are being regulated and just assume that they're being regulated in some kind of ideal way, and from there start getting to emulations that provide insight?
Yeah, yeah. I mean, it seems very plausible that life could be computed on a different substrate other than this brand of molecular biology. And what would that look like if it was different?
Yeah, and what would that look like? What would that look like? Because the thing that's really, really interesting is a lot of what's going on really doesn't look like that the underlying chemistry was the driving factor to the architecture.
But rather it was the relationship between the bits.
Well, it's kind of like your job is to understand how Pac-Man runs on a 6502. Um, Having an oscilloscope isn't that helpful.
Right. The
You know,
important thing
that
is
it
this, what
is
are the abstractions used to generate the game?
it's exactly that. There's exactly that. That it does feel like the only way a system of this complexity could have been designed is if there are very unleaky abstraction barriers. And the whole field of molecular genetics is totally based on bits. It's not at all based on molecules. So the question is there any analog on the metabolism side where you could try to be imagining this as having some clear abstraction barrier that you can build on? Now, the answer to that is it's not obvious that it is. It is possible that the way life works is the abstractions are all leaky, but it's not obvious. It's not obvious in the sense of maybe yes, maybe no. My guess would be that there has got to be some relatively solid abstraction barriers because there's an immensely complex system that works incredibly reliably. The level of reliability kind of means that there must be some higher level descriptions that actually work.
Talking about the reliability of of molecules involved and the
Um,
replication.
it's where I'm sitting now, there's a power line and a squirrel just ran across it. Okay. The complexity of the squirrel maker. Okay. The fact that you could reliably make another squirrel is at some level really surprising.
Yes.
That squirrel is more complicated than the internet.
So what was the statement you made again about the level of reliability
And
being?
right, I prefaced it and I'll preface it again. This is in the wild guess category, not in the facts category. Something that is reliably reproducible probably has sensible abstraction barriers. It's kind of like it's got to be robust to detail. And in order for it to be robust to detail, there must be stuff down at the electrons and protons and bonds level that could be abstracted away from and safely ignored. And it's again, that's a guess. There's no indication that that was true. I was just saying about having that, emulated all computers, as I wouldn't have believed it in that case. And in that case, it worked really, really well because the level of abstraction that the designer in scare quotes worked at was much higher than device physics.
Perhaps an interesting analogy here that is parallel to what you're saying, Brian. In medicine, for example, if you looked at heart rhythms, okay, a healthy person It's fairly regular, but it's not precisely regular. There is some variation, there is some noise in there. And in many medical conditions, that noise disappears to some degree, and the heart rhythm becomes very regular in an abnormal way. And it's that bit of noise that makes the system robust and healthy.
That's a very interesting comment. Because this is free associating. I mentioned at the very, very beginning, What is Life by Schrodinger, right? And he starts it. Have you read it? I
No, I
should.
haven't.
Okay. Just as a caveat, I thought it was brilliant. When I brought it up to Stephen, he thought it was nonsense. So it's very much. He starts the book off with the following question: Why are atoms so small? Okay. And then he said, that's clearly a silly question. The better question is, why does it take so damn many of them to get to a system that asks that question?
That's an interesting perspective.
It's an interesting perspective. Now, what he said next almost is 180 degrees away from you just said. He said, um, deep down, when you get down to the atoms level, it's just noise. Which is what Stephen was claiming in his thermodynamics book. That if you look at each of the individual particles, what you need to do is you need to have enough of them so that the law of large numbers makes the system tractable,
Okay. Yeah.
right? And
It
I,
makes it tolerant to noise.
yes, it makes it tolerant to noise. And the thing that I was saying about being able to reliably reproduce squirrels, that's got to be tolerant to noise. Because if you just yeah, I mentioned this to some other people who may be on this call. I was sitting in a park one day and watching a squirrel chase a pigeon. And somehow I was able to focus in on it, not as the squirrel, not as a creature, but as a machine. And I said, that's a damn good machine. It's able to run up a tree. Imagine like the control electronics for the muscle actuators, right? And um, and it's got a sufficiently good perception system to notice the pigeon and plot a really good path towards it, right? At the same time, it's mechanically very, very efficient. It's energetics. How do you power it? You feed it nuts, right? And if you just think about, that as a machine, it's kind of remarkable. The thing is, and this may be further along to what you were saying, is the way that for a system to be tolerant to noise, it has to be able to ignore it. And to get back to what kind of analysis that we could do with this, is there some stuff that's making our job appear really, really hard that is the noise that the system is going out of its way to be able to ignore?
Well, ignore or capture in a way that's not functionally
Well,
detrimental or something like this. I mean, you have like noise and this sort of genetics from one organism of a species to the next, but what's different is sort of
But isn't
minor
that kind of the same
morphogenic.
thing?
Uh, yeah.
well, what makes this stuff really, really hard to try and emulate is the chemistry of it is messy. There's a lot going on, and we don't really know how to do it. So the question I'm sort of asking, how much of the how much of the chemistry, it's just implementation materials, and how much of the messiness of the chemistry could be treated as ignorable noise?
Mhm.
And it's again, I don't have even the beginnings of an answer to that question. It just seems like it's the sort of thing that repeating the call out at the very beginning, that seems like the sort of thing that would be a good interdisciplinary job between folks who are really conversant with computation and folks who are really conversant with biology. And I don't know if this is being a little bit unfair. I found that on average, the biology people, even people in computational biology, aren't at the level of computational sophistication as a typical Wolfram Institute denizen. Conversely, the typical Wolfram Institute denizen doesn't know, it isn't yet at the starting point to play games with understanding biology. And it seems like there's room for some interdisciplinary work because, because I kind of think it's at some level. So why am I taking a cheap shot at the computational abilities of biologists? They haven't adopted Wolfram language as their tool of choice. Right. So, that's almost an indication that, like, um, some of the stuff that I've looked at are big Python code bases. If you're trying to deal with things in a highly abstract, a Python code bases, you focus. I'm with Stephen in saying you want a computational language rather than a programming language. You know, the flip side, and this was kind of the motivation for what me giving this talk both in the summer and now, is the amount of biology it takes to get to the starting point of this. To say it in both a scary and unscary way, for me, it was only a couple of thousand hours. Okay, which is more than a weekend worth of entertainment, but is tractable for anybody committed to it over the course it's not like needing, um, here, I'll say something that other people could comment on. I find that it's easier to go from zero to being able to read biology professional papers than it is to go from zero to being able to read physics professional papers. You know, that the biology community writes in a language that you just need to take the equivalent of, oh, Bob, you pick a number, 10 university courses, and you could be sufficiently literate to read the literature.
We have a question from Brady on live chat. What would
Yes.
wash out in an emulation, and what would need to be consistent and be contained in an emulation in order to study competition or cooperation over resources?
For the particular case of competition versus cooperation, you could do things at a very, very, very high level, right? Wasn't even there something on the Discord in the last couple of weeks that had some cut at that?
Yeah, I think James posted a video by a YouTuber named Primer, um, where it analyzed resource competition or some sort of voids, essentially.
Yeah, and
So
actually I
yeah,
didn't read
modeling
it, but
agents and modeling their behavior.
yeah, but actually, James, you're online. You must have read the thing that you posted. Is it a right level of what you need to think about and what you could ignore to be studying those things?
Yeah, it's just missing the code.
Okay, that's too bad. I wonder why it's missing the code.
I mean, there might be a repo attached to it, but the point is just to explain the phenomena. They didn't dig into the technical, like how to recreate it for yourself. But that's the best way to understand it is if you actually mess with the code, definitely.
Who wrote this? This is from, yeah, Pedro was saying that to safely ignore details is indeed the art in the craft. I totally agree with that. And, and it's a question, and I think, I was trying to say something along those lines. To try and figure out which details are ignorable is super, super tricky. Because by the way, one of the things that I'd mentioned when I talked to some of you other folks about things around this talk. In Stephen's blog post about engineering in the game of life, he made a distinction between things that looked like they were designed and things that looked like they were found through search. Um, The ATP printing press does not look like it was found through search. And what Stephen said in that blog post is a hallmark of it having been designed. If it's much easier to describe, if you can see how it does what it does, then it's more likely to be designed. And if it was found by search, the simpler description is what it does without being able to look inside of it. Where that particular case of ATP synthase, the electric motor part of it really looks like an electric motor, as would be designed now. And there's a thing in it that looks really like a camshaft. And, the way that camshaft is actually pushing on that last little piece looks biochemically really, really simple.
Yeah, but that begs the question of then what constitutes design?
Um, yeah, yeah, yes, it absolutely, it absolutely does. But well, you got to jump deep into philosophy to get into that, don't you?
Are you familiar with the origins of life research conversation? I'm thinking about Sarah Amari Walker and some of the, yeah, so you're smiling. So I'm
Yeah, I'm
eager to hear.
smiling because at one point, Lee Cronin, at one of the times when I was discussing this, the origins of life question with Stephen, I said, are you familiar with Lee Cronin's work? And that got met with a giant groan. Oh, not him. You know. Now, it turned out that the Not Him was based more on less than ideal personal interactions rather than theory. But so quickly, and you're familiar with the work, I imagine. He's basically
Little
um,
bit, yeah.
what he's saying is, and tell me if this is a correct characterization. A hallmark of design is something that has a high assembly count, which means it's made out of what looks like a lot of sub-assemblies coupled with a high copy number.
Yeah,
So
that's that. Yeah, I had forgotten the terminology, but that's exactly what this conversation has been reminding me of.
yeah, and exactly, if you look at the DNA reproduction mechanism, the energy management mechanism, all of those look like they meet the criteria of having a high assembly count and a high copy number.
But so then are you suggesting that their contribution is just like a classification of stuff we can observe in other ways?
Well, I don't know how much I wanted to get into this one was recorded, but I think we've discussed this amongst subsets of us in the past. What I think is once you've achieved a true von Neumann replicator, it's within the scope of possibility that, well, here, let's try it like this. Let's decide that we wanted to populate the moon. Do you think that we could make self-reproducing creatures self-reproducing, self-powering creatures that could populate a rocky planet without an atmosphere? It would need a different energy system,
Yeah, but we
different.
could do it probably. Yeah.
Okay, good. So if we could make the von Neumann replicator of n plus, if we're von Neumann replicator n, and we could imagine that we could make von Neumann replicator n plus one, could we imagine that we were made by von Neumann replicator n minus one?
Yeah.
Now, somebody should immediately pipe up and say, but that's sidestepping the question rather than answering it because it has to start somewhere. Um, To which the answer would be one only has to imagine that you could make a von Neumann replicator that when you peered inside of it, it didn't look like it was doing anything remarkable. And as a poor analogy, if I look inside of my MacBook Air and I tear it to bits, at all levels, it's remarkable. Okay, it's kind of like there are many, many things in my laptop that could only be made if computers existed. However, if I looked inside of a typical 1960s computer, there's nothing remarkable. Everything there is near to the metal. So one can postulate that it's possible to kick off Darwinian evolution through a von Neumann replicator, but have the substrate be something that doesn't itself make Lee Cronin and Sarah Walker go off saying this is, it could very well be that there's something that doesn't have a high assembly number that is sufficiently powerful to build a von Neumann replicator out of it. And just as a science fiction sense of what I'm trying to say in this, imagine that if we believe Stephen and the principle of computational equivalence is telling us that a hurricane and us are of the same level of computational sophistication, imagine that there are hurricane-like things going on inside of the sun. And the feedback loops get to the point where you do get a spontaneously emerging von Neumann replicator. And these sun creatures say, it's hot in here. Why don't we figure out a different way of living so we could go live somewhere, more hospitable, like the surface of a rock. And they just say to each other, let's ditch nuclear chemistry and move to bond chemistry.
Like that.
Mm.
Well, it's funny though,
Olga is
because
asking about nucleuses of cells. So prokaryotes lack them. Only eukaryotes have these nucleuses. What's the role of that? What's the purpose of a nucleus?
what's the role of, yeah, you know, at the that's the sort of question that's not clear how to give a deep answer. It's almost like eukaryotes at some level look like they're a community, and the nucleus is kind of the shared knowledge of the community.
Mhm.
Right.
Repository for genetic
Plus,
information.
the other poss yeah, repository for genetic information. But this again, this is just complete speculation. The other thing is, in terms of keeping evolution moving fast, sexual reproduction is a really good trick. You need a nucleus for that to make sense.
So what's the chicken and the egg there? Did the nucleus come first or did sexual reproduction come first?
Have you read all of the papers that argue every side of that? Um,
No.
uh, if I had a guess, and this is really, really just a guess, is there's a lot of chicken and egg things. One paper that I read recently that in fact wasn't that bad is the way eukaryotes formed was prokaryotes and megaviruses. Okay, that a megavirus is basically just a big bag of genes.
Yeah.
And in fact, if a megavirus invaded a prokaryote, you'd get something that starts having eukaryotic nature like that. But to the extent that the two big mysteries in origins is the origin of the first von Neumann replicator, and the origin of eukaryotes is probably as big a mystery.
Mm.
What are the clues about eukaryotes? The evidence that mitochondria are symbiotic is quite high. It's the consensus theory now that mitochondria invaded some primitive organism.
Right, right. And then plants, chloroplasts are
And
an
clad,
equivalent
right.
thing. Yeah.
Yes. And what's less consensus, but's being speculated, is the nucleus as well.
Okay.
Okay. And it's being speculated because there are large viruses that people don't understand what they do for a living that look like a nucleus, right? But they don't really, it's not as clear cut as chloroplasts and mitochondria. But in fact, the things that I've been trying to understand, eukaryotes just look like they're too meta and too hard. And I'd be happy to understand prokaryotes and archaea.
Okay. One thing though, the membrane around mitochondria, chloroplasts, and nuclei are lipid bilayers.
Yes.
Okay, but the capsid for viruses are made up of proteins.
Um,
So unless these megaviruses are different,
yeah, no, this is why I was saying is it's not consensus because that is a hole. The hole that I had, actually, the hole I had in the theory, which is literally a hole, the nuclear pore is an incredible structure.
okay.
Okay, and a megavirus isn't likely to have one of those. But if you like, I'll see if I still have a copy of it. I could send you a paper that's talking about, about how the nucleus came from viruses, because that idea, the mitochondria idea, is what it's 150 years old, and it was just brought into the mainstream 50 years ago.
Hmm.
Right. It was Lynn Margulis that actually got everybody to believe it, but it wasn't new to her. That had been an idea that had preceded her by a couple of generations. The nucleus is symbiont, much, much, much newer and much, much less clever. But by the way, to try and talking about discussions like that. Um, there is so much in Origins of Life where you could say, I have no idea. And I think one of the things that's been hobbling Origin of Life research is we've been stalked by the intelligent design people. And you see a lot of people that look like they want to be doing more in origin of life research, but they're afraid of saying they don't know because the moment they don't know, there's a group of people that say, oh, you've got a gap. We have a God to fill it.
Yeah.
Right. Where I actually thought, when I first started having discussions like this with people, people said, oh, you've gone creationist. Right. And um, my answer to that is no self-respecting deity would have required a ATP synthase to do his magic. Right. That the ultimate interesting complexity of it to me is an anti-theistic argument rather than a pro-theistic argument.
Hmm.
Okay. Um, Anybody else?
So, how do you think that the ribosomes got started?
I think they were designed by von Neumann replicator N-1. You see, one of the things I should say that the prevailing, somebody asked that if I read much about origins of life research. I've read a fair bit, and the prevailing, the consensus theory now is the RNA world.
I was about to ask about that. So, the RNA world. What do you
Yeah,
think about that?
yeah. I had the misfortune of before the RNA world being common, having read a book by Christian de Duve, who's a Nobel Prize winning biochemist, who wrote this book saying the RNA world makes no sense. And the argument of the book is RNA is very hard to produce. It degrades quickly. The trouble with RNA is getting the nucleotides together is a hard biochemical problem in a prebiotic environment. Once you get them together, getting them to connect together properly is hard because what they like doing for a living is cross-linking in a way that you don't get mRNA, you get TAR. And if you could get by that, if you could get past that, then they degrade in hours to days. Now, recently in the RNA world, they seem to be claiming that they've got good ways of understanding how you could get strings of up to 100. Um, okay, Um, if you can get strings of up to 100, to get from there to the first DNA-based replicator, you've gone about 1/1000th of the distance. So, um, very weak, very weak circumstantial evidence for there have been a von Neumann replicator N-1. The first is exactly the question that Ellie first brought up. Why are they triples when only 20 of them are used? That to me seemed like there was an earlier version where that was a 16 based. And then the guy said, hey, I can't do this on a 32-bit processor anymore. I need a 64-bit processor. But when you do the conversion, all of your binaries are now deprecated and you have to recompile from source. Right, which
I think it has
I
something to
don't.
do with the ribosome itself, just having a certain valence for these triples and a certain valence for the amino acids that it's coupling with those
Right,
triples,
right.
and
But,
there's
but
some
the thing about it,
ambiguity left in the shapes of those molecules.
but again, here I could repeat the answer I gave before. I think I would buy into that if the redundancy was spread across all three of the base pairs. The fact that all of the redundancy is gathered up in one of them kind of, argues against that.
Yeah, but when DNA, there's the reading frame on DNA, and you can shift by one base pair and generate an entirely different protein. So
No,
it
for
isn't like the every third residue has less information in it, or not every
but
third residue, every third base pair.
aren't you assuming your conclusion? Because, well, the thing that you're saying is a different reason for why all of the redundancy would, to say that, the fact that there's this wobble base pair, okay, is it could be because the advancing mechanism is sloppy, right? You could make that argument. But by
Hmm.
the way, the other things that I thought was even weaker circumstantial evidence. Um, to my eyes, you get RNA by tearing apart DNA. You don't get DNA by, to say that the genetic molecule happened to be something that magically could have been base paired later. That's asking to swallow a lot. To say that in the RNA world happened to pick a fundamental material that base pairs into DNA really nicely. Now, I know that it's a very weak statement, and if I wanted to respond to myself in that, even in the RNA world, there's lots of base pairing going on.
Yeah, because RNA can fold back on itself.
Itself, it can and it does.
Yeah,
Right.
and that probably gives it some stability.
Once it
Um,
folds,
well, what's true in RNA world, RNA, and in proteins, there's a linear sequence that kind of tangles itself into a secondary structure and then folds into a tertiary structure.
Right.
So you get this complicated globular shape. And the end game is the complicated globular shape. And those shapes are ultimately dictated by a linear string.
Hmm.
so what's the purpose? So amino acids are easy to make accidentally, right? You just have these base chemicals there, some heat or
Yep.
thermodynamics, and then they kind of make the
They
amino
come
acids.
together.
They come together.
Yeah,
Now, do those
everywhere
amino
we've
acids come to make chain link on their chains on their
no,
backbones?
yeah, that kind of polymerization doesn't happen by accident spontaneously near as we can tell.
Okay, so you
But
need
the actual
ribosomes to bring the amino acids to make protein.
Yeah, and they do, it's easier to make that happen abiogenically, but it's not easy. Because one of the things that to me was a big issue is if in fact we take as the starting point the bit-spaced replicator, the smallest cut at a bit-spaced replicator that anybody has been able to imagine is about 400K worth of code. And if we were talking about, well, we had a lot of billions of years. We haven't had enough billions years for a 400K program to spontaneously assemble.
But can it be maybe that kind of the existence of the replicator is a consequence of some law of physics that the replicator is minimizing, let's say like local creation of entropy or something like this? Because if you kind of start explaining that everything is rewriting, there is mess being created which you kind of cannot control anymore. That how can you control the mess being created by kind of ordering things next to each other, and what's kind
Well,
of probably the best solution is to create something which just order itself recreates itself. It's some kind of minimum of some optimization problem that basically every possible way would kind of go into the minimum.
um, could I more agree than anything in what you're saying is one of the things that I think, the thing that was what brought us all together is a new kind of science. What that's about is there being rules of organization that we hadn't thought about when we were thinking about physics. One of the things is, and here, this may be too abstract or too stretched a metaphor. The fact that ChatGPT 3 became way cleverer when it got to ChatGPT 4 and nothing changed other than scale. Um, if you would have asked the exact thing that you asked 5 years ago, I would have said, nah, I don't see it. But the spontaneous emergence of skills in LLMs it's another thing that's pointing to there may be some kinds of laws of organization that we just plain don't get.
So it's possible that there could be some mathematical theorem in the future which basically realizes this replicator as some optimal state of organization.
I wouldn't guess that it would be a mathematical theorem because the thing is what it is it's kind of the emergent interaction of huge numbers of some fundamental things. The thing is, here, let me repeat what I said a couple of minutes ago because I didn't say it with enough context. It very much surprised me. So I don't know if you've been playing with ChatGPT as it's gotten better and better. Um, there was a step change between 3 and 4. Um, half of the step change was there at 3.5, but suddenly it got way better. And then you'd start reading papers about what was different between 3 and 3.5 and 4. The answer was just simply more. There was no conceptual advance of any kind. It's just that bigger training set, bigger networks running on faster blah blahs. It was just a quantitative change. And it was a quantitative change that somehow triggered qualitative change. So to get back, something that is very scale dependent isn't obviously something that you can make theorems about.
Mhm.
Yeah, and in a way that's kind of related to your remarks about secondary and tertiary structure of proteins and RNAs and things like that, which short chains by themselves, they can't make very much. But once you get to a certain length, then you can start having these alpha helices or hairpin turns and things like that. So again, it's a question of scale before a new level of organization
For
appears.
sure, for sure. But by the way, since you brought those words up, when I was saying there may be some levels of abstraction that you could ignore the detail. Um, alpha helices, hairpin turns, beta sheets may be that level of organization,
Hmm.
right? Because it does really seem like, um, well, the alpha fold's doing a good job, right? And AlphaFold may have intuited that even though we didn't. Because I don't know if you've have you looked at a lot of these structure diagrams? It's kind of crazy how it really is, it's alpha helo. I don't have a good drawing that I could show people. Maybe the glucogen one that I had. Let me see if it has it.
The
Is
glucagon, yeah, that was just a
yeah.
simple helix.
Yeah, it didn't have any beta sheet nature in it.
No.
No. Um, yeah, and I don't have a different protein thing. It's a shame. Been better to have had one. Um, to just put some more words on what Bob and I were saying to each other, if you have linear sequences of amino acids, the word they use is motifs. Two
Yeah.
motifs that they like folding into is one is a helix, and called alpha because of its direction, I guess.
I don't remember.
Remember, there are beta helices, but people seem to talk. And the other one is beta sheets, where rather than folding into a spiral, it folds into something that's kind of like a sheet.
Well,
And
actually, the sheet is made up where each strand is relatively flat and they come together edge to
uh,
edge,
yeah,
and you make a sheet
and you make a
of
sheet.
strips.
Yes, and understanding protein folding, it seems like the helices and the strips are kind of fundamental things that get used over
And
and
they're all
over,
built from hydrogen bonds.
and they're all built from hydrogen bonds.
Yeah.
And um, well, and half of what's interesting is built from hydrogen bonds, right? Maybe that's a strong statement, but no, it's no, but actually with RNAs, it's not hydrogen bonds,
Yes, it
right?
is between the
It is.
bases. Yes.
Okay,
Yeah,
okay. No,
that's how in DNA and the double helix, the bases form hydrogen bonds to each
no,
other.
yeah, no, that's
And that's
right.
what holds the two strands of the helix
Right, right. No,
together.
but yeah, the thing that I was saying is with RNAs, the base pairing tends to be very small and local.
It
And
is, yes. Yeah.
yes, and most of the structure isn't from the base pairing, but from the frequency of base pairing or something like that.
Right. And the bulges and
Yeah,
things
the
that
bulk.
happen
Yeah,
because it tries
yeah.
to jam too much
Uh,
in too small a space.
Yes, yes.
Yeah.
But to get back, this is all. Uh, the whole von Neumann's whole idea is rather than playing with the shapes, you play with the bit pattern that leads to the shapes.
Hmm.
Right, because this is again, it's repeating. I've said this at least a couple of times up until now. Mutations endure. And to make it mutations that endure, dealing with it in genotypes um, works better for persistence than dealing with it in phenotypes.
Hmm.
There's a question that I have between the role of the genotype and the phenotype. How much of the phenotype is encoded in the DNA and how much of that is up to what the organization of the cell is up to.
Well, okay, a thing that I hadn't said until now, um, in my head, there's this sort of artificial distinction between a kernel and an application. This is like coming directly from from a computer world. The cellular machinery kernel, by definition, is those things that are universally shared across almost all of everything. The application is the stuff that's different. So, um, in fact, if you're talking about evolution, it's all the genotype, unless you believe in what Lamarck said. Though there is a neo-Lamarckism that's coming up based on, people thinking that there are various epigenetic things,
Mhm.
right?
Hmm. Yeah,
But
but some epigenetics does get passed from generation to generation.
yeah, no, but I was just saying, if we would, this is another thing. 15 years ago, if you suggested something like epigenetics, people would scream that Lamarck was wrong. Don't bring that up, right? The recent work with epigenetics is letting a little bit of Lamarckism sneak back in.
Well, it's natural for me because I grew up on Lamarck Drive.
Okay, did
The neighborhood I was in, there was Lamarck, Huxley, Darwin. Those were the street names.
and your parents picked Lamarck over Huxley and Darwin?
I think it had to do with what was available when they wanted to
Okay.
move out of the apartment and buy a house because of the growing family.
No, I was clearly joking, but I'm almost surprised that Lamarck was allowed the same status as the other two.
Yeah.
Okay, for those who didn't spend too much time in their lives with biology, Lamarck was same time as Darwin, a bit earlier.
I don't know. I don't know.
Lamarck felt that that stuff that you did in your lifetime could be passed down to your kids. So it's
Yeah.
kind of like if you did a lot of bodybuilding before you had kids, then your kids would have bigger muscles. And once Darwin came around, the whole idea of what people do in their lifetimes affecting their kids, it was just completely deprecated. Now, um, what epigenetics is saying is there is some stuff that does appear to be passed down genetically, and the mechanism isn't as high level as having exercised a lot. It's instead that in a genome there's various decorations, call it, that
Yeah.
that control expression, and those decorations manage to make it through the gene copying.
Right. Yeah.
The epigenetics being mostly the signals exterior to how DNA is packed in the nucleus, which
No, what epigenetics is in animals and plants, the DNA is usually wrapped around these spools called histones. Okay. And one example of epigenetic information is how tightly or how loosely they're wrapped. And some of that makes it through. A second, the thing that I was calling decorations is there's these little methyl groups that can be added at various points to the genome. And that methylation is another thing that controls regulation. Now, um, there needs to be something like that because the genome of your liver is the same as the genome of your eyeball. Well, not eyeball, but neurons.
mm,
Somehow, neurons need to know that if they're reproducing, they should become neurons, and liver cells need to remember that they're liver cells. So there, actually, Bob, do you know about anything recent? When I last looked at it, this was completely open question with nobody having any compelling theory.
For what? Cell differentiation?
Yes, yeah, cell differentiation.
Yeah.
Okay.
I don't know any details on it, but the little I've read or come across, it's a question of regulation, regulatory events happening in such a way that they're sort of self-perpetuating. Once you start down a certain branch,
Well,
some genes have been turned off and are
um,
not able to be turned back on to get back to basically a stem cell.
yeah, well, I often get myself in trouble by talking to experts in the field and arguing with them about
Well,
expert
I'm not
knowledge.
an expert, but
No, I'm not
yeah.
saying that it's happening this time. I found myself, it was must have been about 30 years ago, just to put a timestamp on it, where I was at a party and a guy there was an expert oncologist. Okay. And what he was doing was 30 years ago, the thing that you just said was the new kid on the block theory. Okay, that at that point, for something for what's cancer is to a first approximation, it's not as simple as this. A cancer cell is a cell that forgot what kind of cell it was, right? That a liver cell that forgot that it was a liver cell will form a tumor, right? And when I talked to this guy, he said that the prevailing theory there was genetic mutation. And his pet theory, which is the one that you just said, is interlocking loops that manage to have some level of equilibrium stability between the loops, right?
Hmm.
And I heard these two things, and I said, I don't really believe either the old theory or the new theory for the following reason. The mechanism the mechanism that is involved has to be a mechanism with resonant frequency of about a decade. And the genetic one is too long, and the metabolic one is too short. Right. So what I thought you needed to find was some kind of mechanism that had the right time frame. And it turns out that the methylations and phosphorylations story do seem to have the right time frame. So I wouldn't be surprised if it turns out that people find that, and since that's kind of the basis of epigenetics these days, right?
Okay.
And again, this is me making stuff up, so not to be taken too seriously. There's no experimental evidence that, nobody's ever looked into this. But but to get back to, Dugan's question is what's in the phenotype? It's basically everything from when you're born until when you die, with the exception of how much your kids resemble you.
Mhm.
Okay, so hang on. Whose comment was this? So it's Petro again. Okay. Um, the thing with Pedro, with what you're saying, and this is it's a sum total of a couple of things that we've discussed already. Clouds, the Cronin-Walker measures of complexity wouldn't have clouds being very complex. Okay, because even though, clouds don't really clone themselves. Um, okay.
Sorry, I'm in a noisy environment, so I'm not
Okay.
speaking
No,
much.
it's an interesting question that and what is it that the thing that I was saying was special, and this is consistent with the Cronin Walker views, is what's different about life than non-life is relatively exact copies of relatively intricate structures. And you don't see that in the weather, to the best of my knowledge. You do see it in crystals. You know, the thing with crystals is they're too exact. There isn't really the variation for, for that to be a super successful genetic material.
Yeah, and the other thing is that for the most part, they're three-dimensional. Um, and I'm not sure how much information you could store in that.
Oh, well, did you read the Carrot Smith book?
No.
Okay. He was just saying that clays aren't fully three-dimensional, and they do have the kind of variation that would allow for them to be possibly genetic material.
I know there's been a lot of speculation about clays being important as a substrate on which some of the early biological chemistry
Right.
started.
Okay. I forgot what his book was called, but it was Karen Smith, and he wrote a book where he basically said he's going to make an argument that's going to go through a series of steps. Nobody's going to agree with all of them, but different people are going to drop off at different points. Okay. So the first point that he made is we were not the first system. We were bootstrapped by a previous system. Okay, I
Yeah.
could go with that. The second is the earliest system was crystals, because crystal growth is providing a good kind of thing. The third step was clays are a kind of thing that have the kind of right kind of persistence and variation to be a genetic material. The fourth thing he said, and this is leading into exactly what you said, one of the things that the clays could have done is they could have started acting as substrates for proteins.
Yeah, an organizational layer on which to build. Yeah.
Yes.
The point I take about crystals, though, is about their replication. They don't reproduce themselves, although they are extremely ordered. They generally come out of solution. Either an aqueous solution, and you get something, like sodium chloride crystals forming, okay,
Yes.
halite, or crystals in magma, for example. You've got a solution there of different metals and ions, and they form different types of minerals, olivine, and I can't remember all the different little ones,
So do you know a lot
quartz and stuff like that.
do you know much about geochemistry?
Not as much as I sound like I might.
Okay, no, because I ran into somebody, I'm going to say I ran into somebody at a bar the other day, and she was doing geochemistry as her
Hmm.
profession. So I said, does anybody ever consider geometabolism as a thing?
In other words, a rock eating another rock or something like that, or
It wouldn't have to,
transforming another rock.
it wouldn't,
Not necessarily. I'm just using rock as a general term
well,
for a mineral.
it's basically, so you've got, a mountain's worth of rock. Um,
Hmm.
Could there be anything going on in very slow motion that looks like it's metabolic pathways?
You're starting to sound Tolkien to me.
Uh, we're
With
talking.
Balrogs under the mountain.
Oh yeah, yeah, yeah.
Yeah.
No, but they move too fast.
Well, I mean, rock is not static.
No, it's not.
It's not static. Ice is not static. It's always
Right.
changing.
And the question that I'm asking is could there be pathways and feedback loops at bigger scales than local?
It seems plausible. I mean, you look at convection currents of magma, things go down and melt and then come back up and then recrystallize,
Right,
Yeah.
get mixed up in different
convection
forms.
currents and magma don't have enough persistence. And, like geology has a lot of persistence. The question is, does it have enough not persistence?
Enough noise.
Well, enough motion, or I'm not quite sure what the right word is.
This motion is just
Enough
a slower time scale.
at a much slower time scale. The question is does it have anything that is of a kind that looks like feedback loops at any level or scale?
I don't know enough geophysics.
I well then when I try to find books about it, it was all I don't know if it was Stephen Jay Gould that talked about the or the earlier phases of science as being stamp collecting, where what people did was a lot of characterization, but not a whole lot about transformations of any kind.
Hmm.
Right, and I don't know, for paleontology specifically, in the early days, there was sort of, for paleontologists, you made your mark by finding something new. So if you found a skeleton that had different features than a different skeleton, you kind of like gave it a new name, but really it could be, a young version and an old version of the same species. Like this is a juvenile, and it, has an adult or sexual dimorphism or something like this, and so they would make new species for each of the um, different types, when really it should be a linear progression of change through a single like lifetime life form. And so there had to be like a going back and looking at over all these skeletons and saying these are really the same species, just different versions of the life cycle.
Hmm.
We have a question on live chat. Brian, do you believe in intelligent design?
Um, okay, can I answer yes and no and yes and no? Okay.
Yeah,
I
sure.
don't believe in anything that anybody's written that refers to what they have written as intelligent design. because the people who are doing intelligent design are, it's essentially, if you look at the people and you look at their friends and you look at that whole field, it's rebranded creationism.
Mhm.
Um, so, do I believe if intelligent design is small I, small d, rather than big I, big D? You know, because like I was saying, is I think whatever happened didn't require any special rule of physics and didn't require anything supernatural. Everything, what I believe is whatever happened happened naturally. Now, I was kind of alluding to this before. Um, somehow, between the Big Bang and 1990s, Mathematica appeared.
Barely.
Well, but what happened?
1989.
Yeah, yeah. Well, I was trying to pick the right date, but somehow, somewhere between the Big Bang and the present, Wolfram language appeared.
Yeah.
Okay. Um, everybody will say, not an issue. Wolfram appeared. Okay. So somewhere between the Big Bang and 1959, Wolfram appeared. Was there another step that would have made it less mysterious? You know, because the thing that I actually believe for sure, to go back to the timeline that I showed early on in the presentation,
Mhm.
there wasn't enough time for the last universal common ancestor to have appeared at the time we thought it appeared.
And that
Right.
was right after the Jack Hill's zircon.
It's you read different books and they keep making the time window shorter and shorter.
Hmm.
People are saying that the last universal common ancestor was like 300 million years after the surface hardened or less. And to think that 300 million years isn't a lot of time, you know, and the other thing that I was sort of arguing is I'm a bit skeptical that the first bit space replicator could have happened in that time. Now, what I thought for years was you could buy an extra billion years by making two bold assumptions, both of which are only marginally motivated. Um, if in fact, if the earliest fossils weren't here but on Mars, um, it gets you an extra billion years or two, for the following reason. Um, when was T zero? It's for Mars, it's when the solar system was created. For
Hmm.
Earth, it's when the moon was created.
Right.
Right. The current theories of the origin of the moon, the earlier theories of the origin of the moon is it happened when the solar system started, but the more modern theories about the origin of the moon could push it, you know, some large amount of time later.
So like
You know, that
a billion year gap between Earth being formed and then the moon hitting it and forming.
well, the thing that this may be too much of a digression. The large impactor theory doesn't make sense as such. The current variant of the large impactor theory is there was a notion that there was a second planet on the same track as us called Theia, and that Earth, you know, Theia eventually caught up to Earth and smashed it. Because if it was if it was something from further away, then it's really unlikely that we would have ended up with such a low eccentricity orbit.
Right.
Right.
Yeah.
You know, Earth's got a very low eccentricity orbit.
Hmm.
So to the extent that there was a large impactor, it should have been a large impactor that didn't change the shape of the orbit.
Right.
And you
And
know,
probably the collision, if it was extra orbital, the collision probably would have resulted in the moon moving far enough away. It wouldn't have been trapped by
yeah.
the Earth's
And,
by the
and,
remnant's gravity.
And there was this, you know, I read this thing a long time ago that said that, you know, there are Lagrange points, and you would expect
Hmm.
junk to appear at one of the Lagrange points, and eventually enough junk would appear to form a second object that would be big enough to matter. And those two objects would never be fully speed linked, so they would eventually get to the point where they would gently collide with each other. And the simulations they're doing about how the moon was formed, the gentle collision, um, seem to make perfect sense. So okay, then we started on Mars. Um, w- Back when I first suggested this to anybody, they thought it was crazy. Since then, ask people, if you ask people who actually study Mars for a living, was there ever life on Mars? You'll get a 60% yes rate, up from about 30 years ago. Because you look at Mars and all of the studies that we're finding is Mars does look like it had lakes or oceans and was a much, much, much more hospitable place in the
Oh, there's no doubt
past
about
than
that.
it.
I mean, it has the biggest canyon in the solar system.
Right. So the other thing is we have a bunch of rocks that came from Mars. And there was this crazy paper in the 90s. I don't know if people are still looking around. There was this one object that came from Mars that they thought it had fossils that looked very lifelike. And since then, we found that life could package itself well enough to easily survive a 50 million mile trip. So it could have been that the early history of life it happened there and then moved here.
Would it not be possible for a particular rock from Earth to go to Mars, then like come back?
It would, but again, the problem that I thought was the problem to be solved was there being not enough time between the formation of the moon and the first fossil.
Yeah.
Right, a very little amount of time happened there, and to the comment of billions of years is it wasn't billions of years, so it wasn't even 1 billion.
Right,
And
right. But
if we look,
okay, wait.
and
So
if
let me
we
get the time frame right again. When, how long ago was Mars formed? The solar system formed, and then when did the moon form?
we should appeal to some of the physicists on the call, okay? Because I don't know the answer to that. Near as I can tell, we think that the solar system formed we date the solar system by dating the Earth. And I don't know if there's anything that dates Mars that isn't just saying, oh, it's the same as here and we can measure here easily. And it's this century that this whole gentle impact with Theia became something that anybody was taking seriously. So I actually don't know. I imagine that there's somebody, one of our friends could give a good answer to that.
Another question is like, do you think some proto life could have formed on Earth and then to be dampened by the formation of the moon, or would the formation of the moon like completely sterilize whatever was trying to happen on Earth at the time?
I thought that we all think that the formation of the moon would have sterilized the planet,
Mm-hmm.
because it just would have turned the surface liquid again.
Yeah, like molten rock.
Yeah. So it's funny though, because maybe the take home from what I'm saying is you're asking a lot of questions. Do I think this or that happened? And I've more convinced myself about things that aren't true than things that are. And like I've convinced myself that the RNA world is probably not true. Right. And so what do you replace? I've totally convinced myself that the God of the gaps means that it's God that did it. I think true or false, that seems like it's a non-answer, that doesn't really help the debate in any way. But, you know,
Right. So your response to the RNA hypothesis was that the RNA molecules are too fleeting,
um,
too too whimsy.
if you,
They fold up too easily, and
so
then they get cut too easily.
I'm just following what, a master biochemist said, right?
Right,
And,
right, right. But basically,
right.
it's like, what would the RNA world give us in terms of like
And,
origins of life?
uh, well, so Deduve
Is
wrote a
it the
paper in
formation of
20.
proteins, like being able to sequence proteins?
Well, okay. The idea is that the RNA world came about when this guy Czech Is that how you pronounce this name? Somebody correct me if I got it wrong. Found that you can have RNA enzymes that are pure RNA that act on RNA.
Mhm.
So the idea that you have molecules that can catalyze reactions on themselves seemed like that's the answer to the origins problem, because then you only need one kind of molecule to bootstrap the thing. Now, it turns out that, and the same Deduve who wrote this book in the 80s that made me not buy into the RNA world when it started, I found a paper of his from 20 years ago where he basically said, well, the RNA world, let's even acknowledge that that happened. You have two big problems, what came before it and what came after it. And let's take the what came after it problem. Let's say that the RNA world actually did happen. It doesn't even begin to explain how we moved to the DNA world. Because as I
or
was,
even the emergence of proteins.
Yeah.
well, they claim it, the RNA world gang does believe that it explains the emergence of proteins. Whether or not I agree, or I guess you don't agree, I don't think it does. There are people that does who think that it does.
Well, so
Because
that's a
the thing
claim,
about
and I mean that whatever the predecessor situation was before, like having proteins or having a
right now,
DNA, you would
in the
need
question
protein
of
synthesis first. I mean, is that
right.
how do you feel about
The
that?
intelligent, right, the intelligent design people who I completely disagree with. It's okay if even if you completely disagree with somebody to say they asked a very good question. They asked the following very good question. All theories of the origin of life are explaining where the hardware came from. Nobody's addressing where the software came from.
Mhm.
And unfortunately, there's enough software there that you can't just brush it off.
Right.
Software being organization, basically, right?
Well, no, being the bits in the genome, right?
Okay.
E. coli, our favorite little creature, has a megabyte of bits in it. Way, way, way beyond that happened it happened by accident. And the Craig Venter folks have minimized it and gotten the genome. They got the genome down to 400 genes.
Hmm.
For E. coli.
But it's artificial and it's not self-sustaining. And even 400 genes is well beyond the hurricane through a junkyard level.
As far as combinatorial explosion complexity is concerned.
Yeah, yeah,
They had done something by modifying the genetic code, didn't they? Where they reduced the number of amino acids needed or
yeah. No,
something.
no, no. What they did is, well, the particular thing they've done more than one thing because they're
Yeah.
a gang of heroes. The particular one that I'm referring to is they took the smallest bacteria they could find, which was a gut bacteria, and then over the course of years, destroyed a gene. And if that kept it from replicating, they put it back, and if it didn't, they'd move on to the next gene. And they blasted away genes down to, and got it down to, again, I think the number was 300 or 400. Now, it turns out that the 300 or 400 that was left, they basically disappeared almost 100% of the metabolism because they're feeding this in a petri dish and they could give it everything that it needs from first
Yeah.
principle.
Yeah,
So
that's right.
the only thing that was left was the information processing piece.
Okay. Yeah.
And even the information processing piece it's kind of like, okay, I don't think I said this earlier. The other reason why I think that something different happened with this, a hallmark of Darwinian evolution, anything that you have something not quite. Okay, whatever you have, you've got something one away and something two away and something three away.
Right.
You don't have sharp peaks. You have, corners of the space, right? The genetic machinery is a sharp peak. There aren't any organisms that have nearly similar genetic machinery.
Even the archaea.
It it's it all um it all is as close as cats and tigers. Okay, it's not as close as cats and mushrooms,
Hmm.
right? In fact, the tRNA shapes that in a mushroom and in you are recognizably the same.
Transcription RNA.
So beg your pardon, the transfer RNA.
Oh, I see.
It's
Yeah.
Yeah, and what it is, and there's just a collection of molecules that are really, really, really universal. And if and most of the really universal stuff are central to the operation of the replicator.
Yeah.
And the ATP ADP currency system is I think that's 100% universal. Yeah, I don't think there's anything alive that doesn't have ATP as an energy currency. Now, the particular ATP synthase molecule, that one was one that's only 99.9% universal because there's some archaea the anaerobes do that a little bit differently.
Right, right.
Right.
I mean, do they
But
still use the same adenosine triphosphate chemical, just a different
yes,
synthase?
they still use the same printing press, but the printing press is less identical. What they do differently is the thing that I was showing before the printing press, the electron transport chain. That's done fairly differently.
Yeah.
But anyway, could I just have another go at saying, you folks, I should try to convince people that there's a lot of fun things to being explored here.
Oh, absolutely.
It's overwhelming.
Well, uh, to be fair, one shouldn't expect to be able to get one's head around what would be at least a 1 year or 2 year university course in the first hour, right?
Right.
And this was more meant to be an ad or a teaser than a collection of answers.
Yes. A good lecture is
Yeah.
one where you leave with more questions than you came in with.
Well, I
So,
feel very tempted. I was doing the calculation of, okay, 1,000 hours. All right, how can I divide that up?
well, I divided it up over about 10 years. And it was sort of, well, it was lucky because at some point in my 20s, I made friends with the guy who liked publishing the current molecular biology text that all the schools were using. So whenever there was a new text, it would just arrive in the mail for me.
Oh,
Oh, nice.
yep.
So that
But
explains it then. Why you're so
Well, no,
yeah.
but one of the things, and I don't know, and it's again somebody. It would be great if this was a comment that I made when I gave the talk the last time. The current molecular biology text starts with chemistry and slowly builds up to molecular biology. I think in my mind, I would understand it better. I don't know with other people if it started with molecular biology that it then unpacked into chemistry.
It would certainly make it easier to begin thinking about the bigger problems.
Well, and it's again, it's a bit hand-wavy to say that the chemistry can be completely ignored. It's probably the case that it could be largely ignored,
Yeah.
right? Because there are things like you correctly pointed out that I didn't mention hydrogen bonding. Hydrogen bonding as a thing is important,
Hmm.
right? And since I said it's important, it would make sense to define it a bit better. So, Dugan, when you were saying that putting that extra phosphate group on was like pushing a magnet on,
Hmm.
it's a similar idea, but a much, much, much weaker magnet, which allows for much more dynamic structures.
Um, I mean, I'm just pointing out that the energy bound up in that bond is one that's kinetic.
It's
Or magnetic. I mean, it's
well, no,
it's bonding of,
what
of
it is.
two molecules.
Yeah, okay. The way that that kind of bond happens is when you have a r squared force and an r to the higher power force. Okay, so what happens is the r to the higher power force, if you get it really close, okay, imagine that it's 1,000 r to the sixth plus 10 r squared.
Yeah.
Okay, when you get it really, really close, it'll it'll stick and it'll stay stuck. But as soon as you push it a little, you know, well, one of them has to be the r squared is repulsive and the r to the sixth is attractive. Hydrogen bonds are a similar thing where there's kind of like two different forces, a short range attractive force and a longer range repulsive force. And that's used to temporarily glue things together in a lot of different important ways.
Mm-hmm. That's what allows geckos to walk on a glass wall.
I didn't know that.
Yeah.
Well, you
It-
could
It's
it-
van der
it-
Waals
it-
process, right?
It's not, it's van der Waals, which has an attractive and a repulsive term.
Yes. Well, the whole thing about having a attractive and repulsive term, it's a good trick that is used at a lot of different scales.
Yeah.
Right. So
Mhm.
should we wrap this up?
Yeah.
Anybody have any final questions, comments?
It's been fascinating.
Well,
Thank you, Brian.
thank you for making it happen. And thank everybody else who's on the call for keeping it entertaining.